Hello, welcome. Good evening.
Good evening, sir.
Audio visual clear.
Yes, sir.
Yes, sir.
Okay, last day we discussed about the Koch postulate, right? So today we're discussing about limitation of Koch postulate. Before we going to into the matter that let's recall Koch postulate. Four rules. Tell me about the Koch postulate. What are the rules? Only human or any other animal? Any other animal. Okay. না ফার্স্টেই দেবে আচ্ছা তার মানে হোয়াট ইজ দ্য পারপাস অফ কজ পস্তুলেট পজিটিভ এজেন্ট পজিটিভ এজেন্ট উইদাউট মাইক্রোবস হতে পারে তো আমরা কোন পজিটিভ এজেন্ট গুলোকে চেক করব হুইচ টাইপ অফ ডিজিজ আর উই চেকিং ইন দ্য কজ পস্তুলেট microbes related. Online student, please answer. Online students answer the questions. Koch postulates different rules. Four rules of the Koch postulate.
So the first one is the suspected microorganism must be present in a diseased organism but absent in healthy organism.
Okay. Next.
Then the suspected organism must be isolated and grown in laboratory culture.
Laboratory condition. Okay.
Then the disease will occur when it is inoculated in the healthy host. Means if mice or guinea pigs.
Same disease or different? Same disease or different disease appears?
Same disease. Same disease.
It should be same disease, right? Okay.
And the last is the same suspected microbe must be present in the disease host.
Okay. So the four postulates are one-liner. Found microbes previously we discussed in lot all of the postulates, but just a one-liner. Found microorganisms in diseased animals and absent in healthy. First one is correct. Diseased organisms should contain the pathogen microbes, pathogenic microbes, and the healthy organisms do not have. And the second postulate must grow in the pure culture. Microbes must grow in pure culture under laboratory condition. Laboratory. And number three, when inoculated these microbes into the healthy host into healthy host, same disease appear. And from the point number 3, 4, number 4 postulates are come. That is, same microbes are found from the dead organism. These are summary of Koch postulate. But after the Koch, when more and more diseases are searched after the Koch, then some limitations are appeared. So limitations of Koch postulate. First limitations are showed by the intracellular pathogens. Limitation number 1 showed by intracellular pathogens. Try to decode the meaning of the word intracellular pathogens. Intracellular pathogens mean, break down words and you got the meaning. Intracellular pathogens. Break it down. Intra and cellular. Intra means inside, cellular means cell. So those pathogens which must grow inside the cell, not outside, only present inside the cell, those pathogens are called, or those organisms are called intracellular pathogens. They only grow inside the host cell. Otherwise, in the laboratory condition, individually, they cannot grow. Yes or no? So those organisms intracellular. Just click down the word intra and cellular. Intra means inside and cellular means cell. So those organisms, intracellular pathogens means those organisms which only grow inside the cell. They are called intracellular pathogens. And these organisms cannot grow individually in the media laboratory condition. They cannot grow. For the growing purpose, they need some helping of host cell. Meaning is clear. What is intracellular pathogens and how it show the limitation in the Koch postulate. So they cannot grow individually. They need the help of host cell. For the growth, they need host. So some examples of intracellular pathogens. The biggest example is virus. Biggest example of intracellular pathogen is viruses, because viruses outside the host cell, it acts as an inanimate and only grow, only reproduce inside the host cell and start to produce disease and some symptoms. So viruses is the primary example that are intracellular pathogen. Clear? So viruses cannot be grown in pure culture, cannot be grown individually. It needs the help of host cell. If you want to isolate the bacteriophage, that is the bacteria virus, you need bacteria for that. Without bacteria, bacteriophage is no existence. So that's why they are not grow individually. They need help of the cell, host cell. That's why they are intracellular. So cannot be pure culture, grown in pure culture. Some bacteria also show intracellular pathogen. Virus are biggest example, but some bacteria, example 2, intracellular pathogen. And a good example is Mycobacterium leprae. Bacterial example which are intracellular pathogen in nature, Mycobacterium leprae. LEPRAE. Mycobacterium leprae cause leprosy in human. Cause leprosy in human. But they cannot grow individually inside the laboratory condition, inside media. We are not successfully grow inside the bacteria, Mycobacterium leprae. So actually, the intracellular pathogens deviate the law number or postulate number 2. Check that. In the Koch postulate clearly mentioned in the postulate number 2, microbes should be grown in pure culture in laboratory. But intracellular pathogens and not go inside the not in the pure culture. They are not go in the pure culture. So they need other host cell for their growth. And the examples of virus, Mycobacterium leprae for the bacteria section. So this limitation actually postulate number 2. Limitation of postulate number 2. Intracellular pathogen. Limitation 1 clear, everybody. Limitation 1 clear. How intracellular pathogen limit the law of Koch postulate? Because they need another host cell for their growth. They cannot be grow pure. Limitation number 2. Prion. Limitation 2 showed by prions. What is prions? Infectious protein agent. infectious protein agent, and that sometimes cause human disease. One good example of human disease that caused by the prion, CR Jacob syndrome, CR Jacob syndrome, and which is analogous to the mad cow disease. I'm sure you heard the term mad cow disease in previous class 11 and 12. Mad cow disease. It is actually caused by prions. No other bacteria, no viruses. They are caused by prions. And the analogous disease, the same disease type that occur in the human is CR Jacob syndrome. So CR Jacob syndrome is a good example of prion, and prion also limit the law of Koch postulate. Limit the postulate of Koch. How? Prions cannot be cultured in the laboratory condition. Prions also cannot culture in laboratory condition. That is, it also disobey, prion also disobey the law of or postulate number 2. Limit the postulate 2 of Koch. Third limitation. Some human disease only occur in the human only. Only occur in human, not other animal. So some diseases that affect the human only affected the human, not other animal in the biodiversity. So how did it limit the law? Because if the human disease only occur in the human, you cannot take human as a model organism. To grow inside, to grow the microbes inside the human and check the disease appeared or not, and from the dead human you check the microbes. Can you do that? Is it ethically correct? Is it ethically correct to take the human as a model organism? Can you? So you cannot take human as model organism, but some of the disease only occurring in the human. So that limit the law of Koch. And the good example is gonorrhea. Gonorrhea caused by Neisseria gonorrhoeae bacteria. Gonorrhea is the disease, human disease, is sexually transmitted disease caused by Neisseria gonorrhoeae. And this Neisseria gonorrhoeae only grow inside the human cells, not any other animals. You cannot take mice or guinea pig here because Neisseria gonorrhoeae do not grow inside the mice or guinea pig or any other animals, only occurring in the human. So that's create problem. So which postulate its limit is the third law. or third postulate, but clearly mentioned when inoculated in a healthy host. That means Koch wants to display the same disease pattern in the healthy host and take the model organism for that. But here in this case, we cannot take any model organism. So this limitation number 3, limit the law postulate of number 3, where the model organism is needed to establish the disease name. So these are some limitations. When more number of diseases are searched and try to link between the microbes involved or not, that time some limitations are appeared, and mainly limitations are come from postulate number 2 and postulate number 3, because postulate number 4 actually the result of postulate number 3. If you fail to do the postulate number 3, the 4 ultimately do not come, and 1 will be correct because the diseased animals, but diseased any humans show the presence of microbes, pathogenic microbes. So postulate number 1 always be correct, but the 2, 3 can be hampered, can be limited in various ways, and some examples are there. Limitation 1, 2, 3. Some microbes we cannot grow in the pure form. Some microbes do not grow in the laboratory condition. Pure form means like example Mycobacterium leprae. They do not grow in pure form. They are intracellular pathogens. And second one, prion. Cannot establish prion inside the laboratory. So difficult to establish. And third one is we cannot take model organisms. Clear? Koch postulates limitation. Online students, clear?
Yes, sir.
Yes, sir.
So to overcome the limitations of Koch postulate, we are now established molecular Koch postulates. So the next concept is molecular Koch postulates. What is molecular? Can you define, or can you just say some things about the molecules or molecular? না মাইক্রোস্কোপিক হবে না, তাহলে মাইক্রোস্কোপি লিখতাম, আমরা মাইক্রোস্কোপিক লিখতাম। ইটস আ মলিকিউলার, correct, nearing to the definition. Molecular. Molecular biology শুনেছো? What is molecular biology? It's the study of genes, means DNA and RNA, right? So molecular Koch postulate, we are trying to identify the genes and the traits, not the total pathogens, whole pathogens. Clear? In the Koch postulate, we try to examine the pathogens presence or not, pathogens grow or not, pathogens produce same disease or not in the healthy host, and same pathogens are recovered from the disease animals or not. Clear? So we trying to deal with the pathogen in the Koch postulate, but the molecular, the new form, the molecular Koch postulate, we are starting about to deal with the genes and various traits of the pathogens. Clear? So in molecular Koch postulates, the actual meaning they are start to identify some traits and they are responsible genes responsible genes for pathogenic microbes. So clearly, they do not want to identify or examine the whole pathogen. And it is true that we cannot deny the concept of central dogma, which is the heart of molecular biology. Concept of central dogma. All of you know the meaning of central dogma because in previous class 11 or 12, as an important concept is there, central dogma. Can you define? Can you guys define what is central dogma?
Transformation, transition, and translation.
That is the process. Clear? Process. But actual mean? Online students, try to answer what is central dogma because in previous class you hear the term central dogma. Anyone? মনোময় তো ট্রাই করছে এখান থেকে, তোমরা ট্রাই করছো না, প্লিজ আনসার।
The process of formation of DNA, I mean DNA to RNA and then RNA to proteins. And RNA can also be again formed to DNA with the help of reverse transcriptase. So this is central dogma.
Which is reverse central dogma from RNA to DNA.
Yes.
So the main process is central dogma, but very few viruses show reverse central dogma, that is from RNA to DNA. But the most of the organisms show the flow of genetic information. So central dogma actually means the flow of genetic information. We all know that genetic information, all genetic information stored in DNA. DNA is the hub of all genetic information. It should be transferred. It should be transferred to what? In the RNA. Then RNA transfer the genetic material or not material, genetic information to protein. The ultimate effect of the all body function is regulated or created by protein. All of the cells body functions are actually maintained by protein. So how does the protein came? How does the protein came? Is protein came from the RNA. And how does the RNA came? It came from the DNA because actual hub material, genetic information material is of DNA. So that it's a regular mean or general mean of central dogma to transfer the genetic information. To transfer the genetic information from DNA to RNA and then RNA to protein. And Monomer telling about the process from DNA to RNA, the process is called transcription. That is the process. But the meaning of central dogma means transformation, changes, or transfer process. Transcription, and here the RNA to protein conversion, translation. So central dogma means transfer of genetic information. That means any protein produced in the cell actually influencing of DNA influencing of RNA first, and then RNAs come from the DNA. Yes or no? Any proteins that is produced inside the cell actually influenced by DNA or RNA. That means genes and genome. The RNA are called genes and the DNA is the genome. So gene and genome has basic difference. Genes means all kinds of RNA and their protein, respective protein, and genome is the material storing material which is DNA. So the proteins actually come from genes and genes are influenced of genome under influence of genome. So genome is DNA and genes are RNA and respective proteins. So in molecular Koch postulate, they try to stick with the concept central dogma. They are not working about the whole pathogen examination. They are start to visualize the genes involved or the traits involved in the pathogenic microbes. Clear? So every pathogenic organisms has some special traits, traits mean characters. Okay, some special traits which are absent in non-pathogenic organism. Do you agree with the statement? Absent in non-pathogenic organism. Hi, Anna. A pathogen causes disease. That means some special traits are there. By that way, it causes disease in the animal or human. Clear? But the non-pathogenic organisms are unable to form disease because the traits are absent. As clear as that. There is no rocket science about how disease are came. Pathogenic organisms contain few genes, contain few traits that clear. So special traits means they are come from special genes. Yes or no?
Yes.
So some special genes you always observe in the pathogenic form, but these genes, correct or not? So this idea is utilized in molecular postulate. This gene can be anything. These genes can be a toxin. These genes can produce toxin. These genes can produce some special structure that involve in the disease formation. These genes can form some enzymes that involve in the disease formation, etc., etc. Can you give an example? Pathogenic and non-pathogenic bacteria, non-pathogenic same bacteria. That is also you studied in class 11, 12. I bet you. Any species, pathogenic, non-pathogenic form bacteria. Same bacterial type, but pathogenic form and non-pathogenic form. Go back to the Griffith experiment. Griffith experiment, Frederick Griffith. What is the experimental organism of Griffiths?
Mice.
What he showed? Experimental organism, mice as well as the bacteria. Mice is the model, but experimental organism is the Streptococcus pneumoniae. and he take two form. One is S form and other is R form. R form is known as R2 and S form is called S3. They define a name S3 and R2. And we know that S3, my Griffith showed that S3 is actually the virulent form or pathogenic form. And the R form is non-pathogenic form. Correct or not? Why it is pathogenic and why R form is non-pathogenic? ক্লাস 12 এ কি পড়েছো সঙ্গীত?
যে S3 আপনার আক্রান্ত করতে পারবে আর
হ্যাঁ কেন? What is the reason behind why S are virulent? Why R is non-virulent? What is the special about S?
আক্রান্ত করতে পারছে
এটা জেনে এসছো।
তারপরে
But in details, what is the special about S form that cause disease pneumonia whenever it goes inside into the human or healthy mice? পড়েছো বাট চলে গেছে। কেসটা ছিল হচ্ছে ক্যাপসুল প্রেজেন্স অর অ্যাবসেন্ট। ক্যাপসুল S bacteria S form contain capsule. R form is non-capsule form. And those are the capsule forms. They are invade easily into the host as well as they can circumvent the immune system, our immune system. Immune system has a special ability of phagocytosis that can kill any kinds of bacteria whenever it inside, and the phagocytosis process will be circumvented by the capsule. When capsule layer is present, phagocytosis cannot be occurred. So S form easily survive in our body, but when R form is inside in our healthy host, then immune system are quickly destroyed by the phagocytosis. So R forms is not present and they cannot produce any kind of disease. But when S form is there, they easily survive because immune system cannot work against the S form as it contains capsule and it produce disease like pneumonia. So the pathogenic bacteria has special structure or not from the non-pathogenic form. I give an example deliberately to show that the pathogenic form has some special structure, has some toxin, has some special things that can produce disease inside when it inside into the host. Clear? So the last statement is all pathogenic organism, every pathogenic organism has some special traits. That could be some toxin, that could be some enzyme, and that could be some special structure. In case of S pneumoniae, it has special structure like capsule, which is absent in non-pathogenic. So this idea is utilized and molecular Koch postulates are given. So molecular Koch postulates have 5 laws or rule. Number 1, virulence trait associated with pathogenic strain. Than non-pathogenic strain. Agree with the statement 1? Every pathogenic strain has some virulent trait. Virulent means disease causing trait, disease producing trait. Every pathogenic organism has some trait, special virulent trait, which are absent, truly absent in non-pathogenic strain. So take two bacteria, same kind of genus species. One is pathogenic and other is non-pathogenic, and check the traits comparison. Compare the traits, and some parts are missing in the non-pathogenic clearly, which are present in pathogenic. That's why they cause disease. Clear? So point number 1, molecular Koch postulate is some virulent strain. Virulent states always associate with the pathogenic strain and absent in non-pathogenic. Second important task, important rule. Number 2, when you inactivate of gene or genes associated with virulency lead to decrease pathogenicity. Second law, agreed or not? Second rule, agreed or not? Inactivate gene or genes, more than one gene also can be involved. Genes involved or associate with the virulency. We early discussed that any traits are produced or appear due to the influence of gene. That is the central dogma concept. Any traits or characters that come from the gene or genome, RNA or DNA genes come from the genes, associated genes. So inactivate those genes particularly. What is here? What is written here? Inactivate them. The genes associated with the virulency, inactivate them, and what will be the outcome? Your expectation. What will be your expectation? Virulency chance will be low, less or low, and found that lead to decreased pathogenicity. So you are confirming that those genes are involved. Those genes are involved in the pathogenicity. Those are the virulent genes. Clear? And next concept, how will you inactivate? The question comes immediately, how will you inactivate the genes? We have different method for that. Inactivation can be done by mutation, clear cut. Induce mutation, induce change. What is mutation means? Changes in the base sequence. DNA contain ATGC base sequence. Change the base sequence. Change the base sequence means change the character of the genes. That means RNA level change. And if RNA level change, protein level ultimately it will change. And this change is harmful and cannot. The proper action will be done. So inactivate those genes which are related to the virulency of the pathogens, and you get the result or outcome low disease pathogenicity, low virulency. And we can inactivate by the means of mutation, induce mutation. Rule number 3, reversion, another way to confirm which genes are involved. Number 3 rule, reversion or replacement of mutated genes with normal wild type, what will happen? Read the statement and answer. Reversion or replacement of mutated gene with normal wild type gene, with the same pathogen, in the same organism. First, you mutated the genes by means of mutation of the particular genes, and the third rule saying about that, reverse or replace those mutated genes with the normal genes in the same pathogen or same organism. What will be the outcome? ফার্স্টেই তো কমেছে, তুমি আবার কি করছো এখানে? ওয়াইল্ড জিন মানে হচ্ছে সেম টাইপ অফ জিন যেটা প্রিভিয়াসে তুমি মিউটেট করলে, দ্যাট জিন ফার্দার ইউ ইনসার্টেড। তো হোয়াট হ্যাপেন্স? Understood my point, online students?
Yes, sir.
So what will the outcome after step 3? Read the statement well. Reversion or replacement of mutated genes. Mutated genes means do not work. That is in the case of step number 2, we induce mutation to deliberately delete it or inactivate the genes. Those are now mutated genes. Mutated genes do not work for the disease progression. Clear? That is clear. Mutated genes. So now mutated genes are replaced with the normal gene. That is genes present in earlier state that cause disease. So now what happened? This organism
Pathogenicity increase.
pathogenicity further, this organism further gain the pathogenicity, yes or no? তোমার একটা ক্যাপাসিটি ছিল, তোমার ক্যাপাসিটিটাকে আমি ফার্স্টে ডিলিট করে দিলাম, তারপর বললাম যে সেম ক্যাপাসিটি আবার তোমাকে দিলাম, তুমি কি করো? আবার তুমি তৈরি করতে পারবে না, সেম প্রডিউস করতে পারবে না। তো দ্যাট জিনস ইনভলভ অর নট, how can you determine? By means of step number 2 and 3. Clearly, first we deleted the genes, first we mutated the genes and check what is the outcome, and it clearly showed that the disease property will be down, pathogenicity, virulency property will down. Clear? And second, these genes, the deleted genes or mutated genes further reverted. Reverted means আবার তোমাকে back করে দেওয়া হবে। So you gain the further property of virulency and start to produce disease. তো এই স্টেপে ডিজিজ ক্যাপাসিটি লুজ করেছে, and থার্ড স্টেপে আবার ব্যাক করবে, কারণ আমি রিপ্লেস করেছি। তো রিপ্লেস উইথ দ্য নর্মাল জিন, মিউটেটেড জিনস আর রিপ্লেসড উইথ নর্মাল জিন, healthy gene that occur in the previous state in the pathogen. So the outcome will be restored pathogenicity, tell me everyone understood or not these 3 steps. It's critical but understandable. If you know the meaning of every word, you can understand easily what the molecular cause postulate try to say, try to describe. So first check what are the properties that differ from virulent and non-virulent strain. Identify those properties. Okay. And this property, how it came from some genes, and check those genes and mutate those genes. Clear? Inactivate those genes and check the outcome of the organism. It can produce disease or not, and clearly show that it does not cause any kind of disease now because the genes are inactivated. Clear? And we further confirm that those genes are involved or not, these genes are further supplemented or given. Clear? And now it checked it restored the property of virulency or not. And we clearly we see that it restored the capacity of virulency. So it will be proved and further proved by statement number 4, 5 or another two rules are there, steps are there to prove further. And the question is how does we replace the reversion? We can reversion the mutated gene from the mutated gene to the normal gene by further inducing mutation. One time mutation cause inactivation, second type of mutation cause reversion. So further mutation
আবার inactive হয়ে যাবে।
না ইনঅ্যাক্টিভটা আবার অ্যাক্টিভ হয়ে যাবে। একটা দেখবে বেঙ্গলিতে ফ্রেজ আছে বিষে বিষে বিষক খায় আছে তো তার মানে কি প্রথম কথা তোমার তো একটা হয়েই গেছে তুমি আবার নাও দেখবে যে মোটামুটি তোমার কিছুটা হলেও গেইন করে যায় তো এরকম প্রপার্টি শো করে ব্যাকটেরিয়া so first you do mutation to inactivate the genes and these genes are further mutated to gain back to the normal type so reversion can be done by further mutation replacement can be done by using genetic engineering genetic engineering another name is RDT recombinant DNA technology so important two tools we are it's actually semester 3 or 4 I think genetic engineering semester 4 is a vast subject vast paper important tools for inactivation or gaining purpose we can use genetic engineering we can use mutation so step number 3 done now go step number 4 the gene involved in virulency should be expressed at some point during infection. Agree or not? Statement number 4. The gene that is involved in the virulency, that means in the previous process, previous steps, we are determining the gene. We are isolating the genes of virulency. Okay. These virulent genes are expressed at the time of infection. Otherwise, how does the disease appear? If the gene is not expressed into the protein or toxin, this expression can form toxin. This expression can form some enzyme. This expression can form capsule structure that make the S strain is virulent. So have to be expressed. Yes or no? So statement number 4 also be correct. If the gene is involved in the virulency during the infection process, it should be expressed into the toxin, it should be expressed into the spatial structure or into the enzymes. Correct? Statement number 4, step number 4 correct? Online students?
Yes, sir.
Yes, sir.
And the last point, number 5. Antibodies directed against the gene product protect the host and eliminate the disease. By eliminating the disease, read the statement number 5 and say it is agreed. You are agreeing this statement. Antibodies. What is the concept of antibodies? What is antibody? Biotic নয়, antibody. Antibiotic, antibody, these two are different term. এটা আমরা এক্সচেঞ্জ ক্রস এক্সচেঞ্জ হয়ে যায় অ্যান্টিবায়োটিক অ্যান্টিবডি অ্যান্টিবডি ডিফারেন্ট অ্যান্টিবায়োটিক ডিফারেন্ট অ্যান্টিবডি our immune system white blood cells that you saying about the white blood cells produce antibody. Antibiotic take from the outside, it's a drug. Our antibody it's from the protein, it's actually a protein and which produce inside our health, our body.
এটা যখন এন প্রোডাক্ট আসছে সেটাকে ওরা নষ্ট করে দিচ্ছে মানে এটা এনিমি ডিজিজ বা যেহেতু ডিটেক্ট করছে অ্যান্টিবায়োটিক
Actual answer is not came. Partially true. Antibody means what is the means of antibody? How does it action against the pathogen? Anybody from the online? What is the role of antibody in our body system?
It affects our immunity.
It
Affects our immunity.
effect মানে ব্যাক রিঅ্যাকশন হবে মানে আমরাই আবার মানে ডিজিজে হয়ে যাব নাকি অ্যান্টিবডি নিয়ে
প্রোটেক্ট করা অ্যান্টিবায়োটিক কাজগুলো আমাদেরকে অনেক সুরক্ষিত করা
হ্যাঁ
Sir, antibiotics are produced against
antibiotics
the
নয়, it's the antibody. 2 points, 2 words are different. Antibiotic and antibody are different. Please make sure it is antibody. Follow.
Sir, antibody are produced by the B lymphocytes
Okay.
against the antigens, and it neutralize the effect of antigens. Means the microbes or the harmful organism that enter our body and cause diseases. It neutralize their effect.
Okay. So the main purpose, exactly the function of antibody to bind with the particular antigen. That is the important. Do you know about the enzyme reaction? Enzyme reaction. Enzyme has particular substrate. Various substrate is there, but it works against a particular substrate. It is very specific in nature. Antibody also doing the same thing. Antibody doing the same thing. It's particular. Various type of antigens are there in our body, but it particularly targeted or bind with the antigen. Clear? So antibody also highly specific. If you say the enzyme is highly specific, as well as antibody also very specific because it choose the particular targeted antigen to bind. Clear? So antibody produced by our immune system that can bind particular antigen if the antibody is against the gene product. Check the statement. Antibody direct against the gene product. Which gene product? That gene product that expressed during the infection time. That gene product. That means it can be toxin. Let's say it's the toxin that cause disease. And during the infection time, that toxins are expressed and cause our health problem. So if the antibodies are produced against the toxin, So those antibodies actually form antitoxin. Yes or no? Antitoxin. So when antitoxin is already prepared, it can protect the host because antitoxin clear the all toxin present in our body system, and we are now protecting from the disease. So that is further proof the gene's activity, the gene's information or gene's role in the pathogen. So these are some steps to identify which type of genes are involved. We here to deal with the genes, not with the whole pathogen. So to check the genes, which genes are involved, to confirm the genes, which genes are actually process in the disease formation, we need to go through these 5 steps. In Koch postulate, there is 4 postulate. Here, 5 steps are needed to confirm the genes involved in the disease. If you confirm that, you do not worry about the pathogen type. Do not identify which pathogens establishment into the host on everything. That means the limitation number 2 or 3. What is the limitation? The growing problem inside the laboratory condition. In this case, no growing needed, no pathogen growing is needed. So limitation overcome. And number second limitation is there is no proper suitable host to study. Here, do not need any host. Check that. Do not need any host. It needs some techniques. It needs some sophisticated technique like mutation, like genetic engineering. And number 4, it could be detected by molecular method of detection method like PCR, ELISA, etc. etc. Some sophisticated techniques are needed. Do not need any kind of host model organism.
এই প্রসেসগুলো করার জন্য তো লাগবে কিছু।
একদমই লাগবে, স্পেশাল ট্রেনিং লাগবে যারা এই প্রসেস জানে অবভিয়াসলি।
মানে এই পদ্ধতিগুলো আবার কারো অ্যাপ্লাই মানে যখন ধরুন এটাকে অ্যাপ্লাই করার জন্য তো মাইস বা কিছু লাগবে,
লাগছে না তো? কোথায় দেখো মাইস আমি ইউজই করিনি কোনো জায়গায়।
যখন এটা তৈরি করা হয়েছিল তখন কি প্রমাণ করার জন্য একটু দেখা যায়
মলিকিউলার কচ পস্তুলেট প্রুফ করার জন্য কোন রকম মাইস মানে হেলদি হিউম্যান হেলদি মডেল অর্গানিজমে ডাজ নট নিড। চেক করো, আমি কোনো জায়গায় লিখিনি আমি মাউস লাগবে বা হেলদি হোস্ট লাগবে, জাস্ট লিখেছি সাম এক্সপেরিমেন্টাল টুলস নিডেড and that tools are highly sophisticated. Yes, that's true, it needs sophisticated techniques, but by that way we can confirm the disease causing genes, and if you confirm the genes, that means the pathogens also produce that product. Clear? Molecular Koch postulates. Clear? Important question. What is the limitation of Koch postulate and how will you overcome the Koch postulate by means of molecular Koch postulates? Clear? All of you clear? Any question regarding molecular Koch postulate or limitation of Koch postulate? Online students, any question, any doubt? Please answer. Any question, any doubt?
No, sir.
Okay. Next concept is pure culture method. Pure culture method is needed during the time of Koch because in the postulate number 2 clearly mentioned the microbes should grow in the pure culture. So during the Koch postulate, during the Koch time, the another concept is needed, another tool is needed to establishment the postulate number 2, that is pure culture method. What is pure culture method? Pure means and impure means
মানে কোনো ইনফেকশন থাকে না, ইম্পিউরিটি থাকে না।
হ্যাঁ আমার কথা রিপিট করে দিল পিওর আর ইমপিওর মিন
আইডিয়াল কন্ডিশনে আর কি।
আইডিয়াল কন্ডিশন অপটিমাম বলতাম পিওর বলেছি only single kind of microbes are present only single kind of microbes present in your sample, that is your pure culture, pure organism, because no contamination is there. Impure means contamination. Impure food product means your food product contain other things, extragenous things, right? So pure culture methods is another concept during the time of Koch, another important tool. So pure culture means a culture containing only one type of microorganism. Only one type of microorganism. That is a pure culture. And it's regarding the opposite what is contaminated culture. That means more and more mixed type of microbes are grown in the culture. That is contaminated culture. So a pure culture is always needed to establish the Koch postulate. Clear? Do you think this pure culture can you observe in the liquid medium? Which is the better mode to obtain pure culture, liquid medium or solid medium? Can you guess what is the better medium purpose to obtain pure culture? Means single containing cell, single organism containing culture. Reason? Monomoy answer solid culture. Agree with or you differ? What is the better mode? Pure culture method, better mode of culturing solid or liquid medium? Give some point. Why? What is the better? আনসার করো আদারওয়াইজ গ্রো করবে না নলেজ আমি বলে দেবো এক্ষুনি বাট গ্রো করবে না জাস্ট থিঙ্ক অ্যান্ড ট্রাই টু আনসার হোয়াট ইজ বেটার মোড হোয়াট ক্যান বি দ্য বেটার মোড ফর পিওর কালচার আইসোলেশন
Sir, solid medium, I think.
okay why you think it's a solid medium?
Sir, because the molecules are tightly packed in solid medium, so there will be less contamination.
Good point. Jahnavi said about in solid culture there is a compaction, high level of compaction about the molecules which form the base. That's why the contamination chance will be less. Correct point. So what problem associated with the liquid? Number one, cell aggregation. When cells grow inside the liquid media, grow vigorously, quickly, not quickly, vigorously. That's why they start to form clump. And clumping cell contain mixed type of population, not one type population. Number one problem about liquid media to obtain pure culture. Number two, the high chance of contamination. Clear? Whereas in the solid media, when cells are formed, when cells are grow, they form colony. Cells after growth inside the solid form colony. Introductory class, in the introductory class, we discuss about how to grow. There is a potential problem to grow microbes. So where I discuss about the importance of medium, solid medium importance. Inside the solid medium, cells are grow and form colony. Okay? But in liquid media, cells are start to aggregate and dispersely they are present. Does not form a colony. They grow vigorously but dispersed. But here they try to form colony. And what colony means? What does it means? Colony. Colony means a single colony contains single type of organism, and that from the pure culture. Correct or not? A single colony, a single colony contain only one type of organism.
Sir, in liquid medium, they also form colony, but they get dispersed.
No, they do not form colony inside the liquid. They are form aggregate, but it is dispersed form and it looks like hazy. When the microbes grow, it looks hazy, but no colony formation is there. But in solid, there is a colony, typical colony. Check the difference between liquid medium growth and solid medium growth. Liquid medium growth, microbes grow and it haziness or viscous the liquid, viscous the medium, and that conformation of growth is occurred. But in case of solids, they form this is one colony, another colony, and this colony, this single colony contains just single type of cell. Because where in this case, when you grow bacteria inside the solid medium, there is a single bacteria, and single bacteria consume all the nutrients in the periphery and start to form millions of cells and grow into the colony. So in colony, no mixed type of cells are formed. Bolo?
No, sir, my doubt is
Okay.
clear now.
Okay. So this colony contain only one type of microorganisms, one type of organism, and that forms pure culture, which is essential to establish Koch postulate. So clearly, solid medium is better than liquid medium from that view. Yes or no?
Yes, sir.
So next, the challenging task to solidifying the medium. How can we solidify a medium, liquid medium into solid medium? That's the challenging task. And Koch actually using liquid to solid medium tried various things, Robert Koch. But some of the methods are failed, and ultimately he used first he used gelatin to solidify. Then he used agar, popular material. Nowadays we are using in the laboratory condition agar. But the agar discovered by agar can be used as a solidifying agent discovered by Robert Koch. But actual the history came not the Robert Koch. The Robert Koch colleague's wife actually helped to decipher the to use the agar. So first, Robert Koch start to use gelatin to solidify the medium, but gelatin has some drawbacks. Gelatin has some drawbacks. Number one, gelatin can easily degradable. Gelatin easily degradable. Actually, gelatin, any solidifying medium contain a base, right? Solidifying medium contain a solidifying agent. Solidifying agent act as a base. If base is degraded, so how it will be solidified? So many bacteria can easily destroy the gelatin by production of gelatinase enzyme. So drawbacks of gelatin using solidifying medium, solidifying agent. Number one, easily destroyable by gelatinase enzyme. And another potential problem using gelatin as a solidifying agent, it melted down above 28 degrees centigrade. It melted down above 28 degrees centigrade. प्राय room temperature ही melt हो जाए. Start to melt from 28. So any disease causing microbes, human disease causing microbes, can you prepare solidifying medium by using gelatin? Because many microbes that cause human disease are grow at physiological temperature. Our physiological temperature 37. Our physiological temperature 37. So many bacteria cannot grow inside the solid medium, inside the gelatin containing medium, because gelatin at 37 it is liquid. So what is the benefit of using gelatin? So the same ही होएगा. Previous liquid medium and gelatin containing medium is at 37 is same. So no use of gelatin after 28 degrees centigrade. It can be used at the cold, at refrigeration temperature, but after 28 it will be start melted down. So Robert Koch start using gelatin at first, but later it replaced with agar. And agar is suggested by his colleague's wife. Colleague's wife name Fanny Hess. Agar is suggested by Robert Koch's colleague name is Walter Hess, and his wife Fanny Hess recommend to use agar as a solidifying agent to Robert Koch. Why she suggests to use agar? Because she found that Fanny Hess, she used agar to make jelly. Jelly, jam, etc. And she showed that it is a good solidifying agent. It makes the structure solid or gel-like form. So when agar is used by the Robert Koch, he found some advantages. Question can be formed from that. What is the advantages of agar over gelatin? When Robert Koch used agar, he found some advantage. What is the advantage? Number one, agar cannot be degraded. Most of the bacteria, gelatin can be degraded by the bacteria by production of gelatinase, but agar cannot be degraded. That is the first advantages. By most of the bacteria. So it acts as a good support system for any most of the culturable bacteria. Number 1 point. And number 2 point, which is the problem in the gelatin, it melted down after 28, but agar melts after 100 degrees centigrade. At around 100 degrees centigrade, that means boiling temperature. Agar only be melted at above or at around 100 degrees centigrade, which will be beneficial. And another beautiful features of the agar, when it is melted, once it is melted, it cannot be further solidified before 50 degrees. Once it is melted, it cannot solidify before 50 degrees. Another beautiful features of agar to utilize as a solidifying agent. So 2-3 features make the agar suitable for solidifying agent. Number one, it cannot be utilized by most of the bacteria. Number two, above 100 degree, it will be melted down, not at the 28 or 38 degrees centigrade. So most of the bacteria can grow as a solidifying agent of agar. Clear? And number three, another important thing, once it is melted, it cannot be solidified before 50 degree. So that eliminates to handle the boiling agar. মানে তোমাকে 100 ডিগ্রিতে তোমাকে ইনকুলেশন দিতে হবে এরকম কোনো ব্যাপার না, তুমি আস্তে আস্তে তুমি 50 ডিগ্রির কাছাকাছি আসলেও তুমি নিতে পারবে, মানে কোনো স্পেশাল নো নিড এনি ফ্যান্সি ইনস্ট্রুমেন্ট টু হোল্ড দ্য agar. You can hold it with the normal naked hand. তোমাকে যখন করতে দেবে ল্যাবরেটরিতে তুমি নেকেড হ্যান্ডে ধরতে পারবে লিকুইড agarকে, বিকজ ইটস অ্যাবাউট 50-55, ডাজন্ট ম্যাটার, কিচ্ছু হবে না। Clear? So another beautiful feature that make agar suitable, that it eliminates to hold at 100 degrees centigrade, because 50 degree above it will be liquid. Before 50 degree, that means 40-45, it will be solidified. So it eliminates to hold the liquid at above 100 degree. This point is clear or not? Third point is point number 3. Why agar is better substitute of gelatin. And today in modern condition, we use agar as a solidifying agent. So today's class is done. Just check it and confirm everything is okay or not. So in today's class, we discuss about the limitation and how the limitation is overcome by means of molecular Koch postulates. And during the Koch postulate, the one important thing was pure culture formation, that is production of solid culture to identify the colony, or pure method, pure culture. So that is the concept pure culture, and how it will define, how it will produce pure culture, and what is the better agent of solidifying agent, better solidifying agent, agar or gelatin. First of all, they are using gelatin, but there is many drawbacks. That's why Robert Koch choose to use agar, which actually suggested by Fanny's colleagues. তো কিভাবে কানেকশন হয় দেখো, যখন একটা ডিসকভারি হয় কিভাবে কানেক্টেড অল থিংস, তো একটা সায়েন্টিস্ট মানে স্মল স্মল থিং গুলোকে নোটিস করে। দেখো রবার্ট কচ চাইলে ফ্যানিংস এর কথা হেসে উড়িয়ে দিতে পারতো, জাস্ট মক করে ছেড়ে দিতে পারতো, বাট অ্যাজ আ সায়েন্টিস্ট ইউ হ্যাভ টু বি ওপেন মাইন্ডেড, সমস্ত কিছু নিতে হবে। তুমি যদি রোবাস্ট হয়ে যাও, নিজেকে বিশাল ভাবো শুরু করো, আলটিমেটলি কিচ্ছু হবে না, তুমি একটা জায়গায় স্টিক থাকবে। এই যে জিলাটিন ইউজ করে যাচ্ছিল, জিলাটিন ইউজ করলে আলটিমেটলি নো রেজাল্ট। ম্যাক্সিমাম মাইক্রোবস ডু নট গ্রো, only below 20 degree centigrade temperature microbes, though are preferable to grow below 28, they are only grow. So majority microbes they are missed to grow the solid. Clear? So have to be open minded all the things all the time. Done? Any question? Clear?